Question number 176. Choose the organism whose natural product is used as a clot buster for removing thrombus. And the options are Entamoeba coli, Streptococcus species, Methanococcus, and Vibrio cholera. And the correct answer to this question is option number 2, Streptococcus species. Because streptokinase is produced from Streptococcus species or Streptococcus bacterium and modified by genetic engineering and is used as clot buster for removing thrombus from blood vessels of patients. Let's discuss rest of the options. The next option is Entamoeba coli, and this is an incorrect option. Entamoeba coli is a non-pathogenic species of Entamoeba and is an incorrect option. The next option is Methanococcus, and this is also an incorrect option. Methanococcus belongs to the kingdom Archaea. It is a thermophilic organism and creates abundance of methane gas, and so is an incorrect option. And the last option is Vibrio cholera, and this is also an incorrect option. Vibrio cholera causes acute diarrheal infection called cholera. So the correct answer to this question is option number 2. Let's proceed to the next question. Question number 177. Which of the following enzyme is thermally stable? And the options are pancreatic lipase, Taq polymerase, gastric protease, and EcoR1. And the correct answer to this question is option number 2, Taq polymerase. Taq polymerase is an enzyme which is obtained from bacterium Thermus aquaticus. It can survive temperature up to 95 degrees centigrade. It is a type of thermostable DNA polymerase. Let's discuss rest of the options. The next option is pancreatic lipase and gastric protease, and these are the incorrect options because these are the digestive enzymes and they are not thermally stable. And the next option is EcoR1, and this is also an incorrect option. EcoR1 is a type 2 restriction endonuclease and is not a thermostable enzyme. So the correct answer to this question is option number 2. Let's proceed to the next question. Question number 178. Which of the following is a source of energy during a PCR? And the options are ATP, DNTPs, DNA polymerase, and thermal cycler. And the correct answer to this question is option number 2, DNTPs, and not ATP. Because ATP is not needed, as deoxyribonucleotide trisephosphates can themselves provide energy. Whereas ATP, which is the energy currency of cell, is an incorrect option. DNA polymerase is an incorrect option. DNA polymerase is used in extension step of PCR and it is used to form a new DNA strand. And the next option, thermal cycler, is also an incorrect option. Thermal cycler or PCR machine raises and lowers the temperature of the block in discrete and pre-programmed steps. And this is also an incorrect option. So the correct answer to this question is option number 2. Let's discuss our next question. Question number 179. There is a restriction endonuclease called EcoR1. What does alphabet R in it stands for? And the options are genus, species, strain, and order in which the enzyme was isolated from the source bacteria. And we know that the convention for naming these enzymes is that the first letter comes from the name of the genus. So first letter represents genus, second letter represents species, third letter represents strain, so fourth letter represents order in which enzyme was isolated. This means R is derived from the name of strain rough, which means the correct answer to this question is option number 3. Whereas genus is an incorrect option. E represents genus. In this nomenclature, E represents genus. Option number 2, species, is an incorrect option. CO in this name represents species. And fourth option is also an incorrect option because this Roman number 1 represents order in which the enzyme was isolated from the source bacteria. So the correct answer to this question is option number 3. Let's discuss our next question. Question number 180. Insertion of gene of interest in PBR322 at which restriction enzyme site results in susceptibility to ampicillin in recombinant E. coli? And the options are PVU2, PST1, BAM-H1 and CLAV1. And we can see that in this ampicillin resistance gene, we have site for PVU1 and PST1. So PST1 lies in the coding sequence of ampicillin resistance gene, and insertion of gene of interest at this restriction site, at these restriction sites, can lead to insertional inactivation of this antibiotic resistance gene, thereby leading to susceptibility to ampicillin in recombinant E. coli. So the correct answer to this question is option number 2, PST1. Whereas rest of the options are incorrect because PVU2, as you can see here, PVU2 lies in coding sequence of ROPE, and ROPE codes for the proteins involved in replication of the plasmid, and insertion of gene of interest at this site will not result in susceptibility to ampicillin. The next option is BAM-H1, and this is an incorrect option because insertion of gene of interest at BAM-H1 site will result in susceptibility to tetracycline and not to ampicillin. And the third option is CLAV1, and this is an incorrect option because insertion of gene of interest at CLAV1 will not result in susceptibility to either tetracycline or ampicillin. So the correct answer to this question is option number 2. So with this question, I would like to end up the discussion of this AIATS paper. I hope all your doubts and queries have been solved regarding this paper. Thank you very much for watching and all the best.